Sorry, I'm a little sick today. That means that whatever virus or bacteria that reached my bloodstream, it went past all my innate immune defenses, whether that be my skin, my stomach acid, or my white blood cells. Sorry. But a good thing is that I have specific immunity developing right now, so hopefully I won't have a problem with it next time. So let's get started onto the whiteboard. So right here I have a pathogen, and this pathogen is drawn in blue. Off of those pathogens I have small little red dots, and those are called antigens. So the point of immunity is for the body to recognize these new pathogens so then they can learn how to fight it off. Generally, when a virus or a bacteria is introduced, there is a general immune response, which means that there's going to be something called a macrophage. And this macrophage is going to engulf the pathogen through a process called phagocytosis. Phagocytosis is a type of endocytosis. It's going to engulf it, and then it's going to break it up into little pieces along with the help of lysosome. And these little fragments are going to, so the macrophage has something called MHC2 that's going to present the pathogen. And since the macrophage is presenting the antigen, we'll call it an antigen presenting cell. It's going to attach to a T helper cell. T helper cells and macrophages are both different types of white blood cells. The T helper cell is going to bind to the MHC2 with its specialized protein called CD4. And with that, it's going to clone the T helper cell, because the more T helper cells we have, the better it is for the immune response to fight off the pathogen. The T helper cell will then go into either humoral response or cell-mediated response. So these T helper cells are going to release a chemical called cytokines. So cyto means cell and kine means to move. So they're going to move these chemicals to a cell. And it's going to move to B cells in the humoral response. B cells, we know, mature in the bone marrow and they make antibodies. While T cells are also made in the bone marrow, they are matured in the thymus. And so the B cells will then recognize the antibodies that are attached to the pathogen. This is the pathogen. It's going to have the antibodies attached to it. And that's when they know that it's time to strike. So these B cells can turn into memory B cells, because if this virus or pathogen were to come into the bloodstream again, the body will recognize it and attack it and have a faster response. Let's talk about the cell-mediated response. The T helper cells can also go and signal cytokines to bring in the cytotoxic T cells. And these cytotoxic T cells deal with pathogens that are inside a cell. It'd be hard to get the pathogen and kill it, but we need the help of cytotoxic T cells. These cytotoxic T cells are going to bind to the cell membrane of the infected cell, and then it's going to release its enzymes to break through the cell membrane and then eventually kill the whole cell, something called apoptosis. So cell mediated happens inside infected cells. Okay, so let's review. Here's a chart on humoral and cellular immunity. So I have the differences between each. Humoral response occurs primarily in B cells, and then cellular immunity occurs with T cells, whether it be helper T cells or cytotoxic T cells. And humoral immunity uses antibodies to either neutralize or mark them for destruction, while cellular immunity directly attacks and kills the cancerous or tumor cells. And with humoral immunity, as we saw, it targets pathogens that are outside of the cell, and cellular immunity targets pathogens that are within a cell. Immunity has a faster response, while cellular immunity is a slower response.